Alrighty guys, so this is going to be the overview video for cell signaling, and I suggest that you watch this one before the other three. So the chapter starts off with, you've got this video of a gazelle that's being chased by a cheetah, and then you have a big idea question that says, what cell signaling process causes this fight or flight response in the gazelle? So the overarching phenomenon basically is saying, okay, we have this gazelle being chased by a cheetah, and so how does this gazelle send signals to all of its body that it needs to do this, quote, fight or flight response? It starts increasing its breathing rate, it starts increasing its heart rate, it starts releasing sugars for the energy to be able to run away from the cheetah, but how does its body send those signals to do that? And the idea is very similar for us, right? Like if you've ever been nervous or scared about anything, maybe you've been nervous about taking an NAP bio test, or you've been nervous about giving a class presentation or getting in trouble or whatever, right? Then how does your body send those signals to you to start sweating or to start running away if you're that scared, okay? So that's kind of the big idea behind the chapter. So let's start a little bit smaller because cell communication, first we're going to start talking about the different types of communication between cells, and then we're going to go through more specific mechanisms of once the cell receives the signal, what do they do with it, and so on. So first let's start with how unicellular organisms like yeast and bacteria communicate. So unicellular organisms can communicate with others of the same species in order to do many different things, like signaling whether there's availability for food. They can communicate where they identify their mating types. They can detect others for coordinated behavior, that kind of thing. So one example here is with the yeast. And this picture's in your textbook. But what happens is, so yeast is a unicellular fungus, and it reproduces sexually sexually and chemically by emitting specific chemicals called factors. And so how they reproduce is that they'll usually exchange some genetics with each other. And in this diagram, yeast, unlike us, they don't really have a gender per se. So we don't have, males and females and things like that. They just have a different mating type. So this would be the mating type A, and this is mating type alpha. And so what's happening is that if they're different mating types, these yeast cells that meet up with each other, then they will send their factors. So yeast cell will send a factor, and then the mating type alpha will send its alpha factor, and so then they will send those signals to each other, and then they will fuse together, and then that creates what we call a diploid nuclei, which means that it has the genetic information from both mating types. And so once it becomes a diploid nuclei, then it will divide and it will form a new organism with genes from both of those mating types. Okay, so therefore, the yeast can really commonly communicate by detecting cells of the opposite mating types, and then exchange their factors, their mating factors that way, and then, divide and reproduce, okay? Another one though, and that's why this is like really not so enticing picture of someone's mouth, and particularly their teeth. And particularly it's the plaque on the teeth. There's another type of communication with unicellular organisms like bacteria. And that's what we call quorum sensing. So quorum sensing, it's spelled Quorum sensing. And it's something that bacteria use to communicate. So bacteria can detect the presence of other bacteria, and when they do that, they will group together or aggregate, and they'll form a hard film called a biofilm, and that's kind of what this is. And And it's really hard to get rid of it. And so that's why, you know, it's super important to brush your teeth every morning and every night, because if you're not brushing your teeth well enough, then the bacteria start aggregating and more and more and more to where it becomes really hard plaque. And that's why when you go to the dentist office sometimes and they clean your teeth, and, you know, if they've ever been like picking really, really hard to try to get the plaque off, that's why, because it's really super hard to get off. So again, the definition, so to speak, of what quorum sensing is, bacteria will secrete small molecules in the form of a chemical that will be detected by the other bacteria. And then once they've done that, then they'll start to sense that the population around them is super dense, and it's dense enough that they'll start performing a group behavior, and that group behavior is called quorum sensing, right? So, and that's what we ultimately call a biofilm. And so it's a way, it's kind of a protective behavior for the bacteria, but it's, well, obviously annoying to people like us with plaque and stuff like that, right? So, okay, we're going to move on. That's the bacteria. So we'll move on to, here we go, this slide. So we can classify cell signaling based on distance. And so earlier we talked about unicellular organisms. We're going to move on to multicellular organisms with cell communication. And so with multicellular organisms, they can they can communicate similarly to unicellular organisms, but they will send their chemical messengers. They can either be sent short distances or they can be sent long distances to target cells. And so this slide is a little confusing the way that it's organized. So let me just kind of go through it in a more simple way. One way of cell signaling in a multicellular organism is signaling between adjacent cells, and that's what we would call, and that's this picture right here, and that's what we would call juxtacrine signaling. So that would be like sending signals through the gap junctions between animal cells, or sending signals between the plasmodesmata between plant cells. So really in this case you're sending the signal just directly into the cytoplasm. And the other form of juxtacrine signaling is going to be cell-to-cell recognition, where you have neighboring cells that have cell surface receptors. So the signal will be sent directly to the neighboring cell. And usually this kind of signaling is really important in embryonic development. And it's important in a process, it's called embryonic induction. And what that does is when you're first been fertilized as a zygote in the womb, and then the cells start dividing and dividing and dividing and dividing, and so as they're dividing and they're starting off as stem cells and then they want to become differentiated. Well, in embryonic development, as they're becoming differentiated, they will start sending signals to each other to say, hey, you're going to be an eye, so let's divide and make all the eye cells, or hey, you're going to be a liver, like let's divide and make all the liver cells. So that's kind of the simplified way of explaining embryonic induction. But that embryonic induction uses this kind of a signaling. And then the second way of signaling based off of distance is what we call local signaling, and that would be paracrine signaling, or sometimes synaptic signaling. Okay, so paracrine signaling is usually for when the cells want to mitotically divide. So that's why we have growth factors are usually the ligands involved in paracrine signaling, and then synaptic signaling is for neurons, okay, when you want to fire a neuron. So in this case, the ligand would be the neurotransmitter, and then those neurotransmitters are released by the neurons across the synapse into the next nerve cell through exocytosis. Okay, and then finally we have long distance signaling, which is over here. Long distance signaling, usually we refer to it as endocrine signaling, and anytime you hear the word endocrine, just like our endocrine system, think of hormones. So long distance signaling is hormones, and the reason why it's long distance is because hormones must travel through the bloodstream. Okay, and in this case our hormone is our ligand. So in order to reach the target cell, the hormone has to travel through the bloodstream. So for example, gland cells will will secrete the hormone, and then that hormone will go into the bloodstream, and then it will travel all the way to the target cell and do what it needs to do. So, let's say our pancreas is making insulin, and then that insulin will travel through the bloodstream into the liver to where it will be used. Okay, so that's kind of a little bit of an overview of the signaling. So let's go ahead and go through the 3 stages of cell signaling. So the first stage is reception, right, and that's where the signal molecules bind to the receptor protein on the cell surface, or they can bind to the receptor protein intracellularly, and we'll go over that, okay? But only ligands that pass the cell membrane are, well, let me back up. The only ligands that can pass the cell membrane and bind to something that's intracellular are ligands that are hydrophobic or lipids. Okay, so most of our ligands are hormones actually. So hormones, that would be something like testosterone or estrogen. A lot of the sex hormones are lipids, okay? Or steroids can pass through as well. So something that is hydrophobic and made of primarily lipids will be able to pass through the membrane and bind to an intracellular receptor protein, okay? But other than that, like the other hormones that are proteins, like insulin or something like that, those will bind to the receptor proteins on the surface of the cell, okay? And then the process of transduction, which happens intracellularly. So transduction is really you're converting, well, it's the conversion of a signal to a form that causes a cell response. And in other words, you're transducing a signal to a form that can cause some sort of a response by the cell. And it involves many, many, many steps and many different molecules, and one reason for that is so that we can amplify the signal and then ultimately fine-tune it as well. And then the response phase, there's just a variety of different responses that are possible. I mean, most likely it's turning on a gene to make a protein, or we're activating an enzyme, or we're doing something with cell structure, or we're causing the cell to divide, or we're stopping the cell from dividing. But I mean, there's multiple different responses that can happen. Okay, so we're going to move on to reception. Brief overview here, we have 3 different types of receptor proteins, right? So the GPCR, usually this is where, like for example, most of our bacteria, if you have a bacterial infection, usually those are caused by the bacteria will release some sort of a toxin that interferes with the GPCR. So most of the medicines that counteract bacterial infections usually work to restore the function of this protein, okay? And then receptor tyrosine kinases, why are those important? They're important mostly because of cancer. So they're famous due to the fact that they often trigger like an enormous amount of growth and cell division. So in other words, they are usually, a malfunction in a receptor tyrosine kinase is usually what causes cancer. So if there's a mutation, for example, or something else wrong with it, then it will send a constant signal to the cell to continue to divide, and ultimately that will create a tumor, which then becomes the cancer, right? So for example, a lot of breast cancer research today, that's what they try to do, is they try to disable this mechanism in the receptor tyrosine kinases that when they get mutated, they try to disable that so that in a way the response is normal instead of inciting more and more division. So that's one thing that they're trying to come up with. And then the ligand gated, obviously we've been over these and how they work, through cell transport, but ligand gated channels is usually how the neurons will function. So the ligand will be your neurotransmitter, and then it will bind to the ligand-gated ion channel, and then it will allow sodium ions to move through, and then when those sodium ions move through, that will start the action potential, okay? And then the intracellular one, so this is where the receptor protein is on the inside of the cell, and again, the ligand in this case has to be some sort of a hydrophobic hormone like testosterone. So it will move through the membrane, bind to the receptor protein inside. Then this will actually travel through the nuclear membrane and then bind to DNA usually, and it will often turn on some sort of a gene and make a new protein and things like that. So remember that hormones, when they're sent, though, they're usually sent to all of the cells in the body, right? But only the cells that have the particular receptor protein that the hormone wants to bind to will elicit the response. So in that case, all the cell signaling pathways are extremely, extremely specific. Okay, and so we'll end this overview, part 1, and then we'll make a part 2.